Tobacco use is the leading cause of preventable disease, disability, and death in the United States. Nicotine is the highly addictive chemical found in tobacco products. Nearly 40 million adults smoke cigarettes, and about 4.7 million middle school and high school students have used at least one tobacco product, including e-cigarettes. Society has glorified using tobacco products and has made it quote-unquote cool to use. Impressionable teenagers and curious young adults that are socially using nicotine run the risk of becoming dependent on nicotine. Nicotine dependence is the ability of the addictive substance of nicotine to affect your mood, and you feel as though you cannot go without it. The harmful chemicals in tobacco products have an impact on all the cells in your body, but can seriously affect the heart, lung, and brain cells tremendously. Nicotine impacts your dopamine levels, the neurotransmitters in your brain, and your lung and heart function. Both genetics and your environment can affect whether or not you will become dependent on nicotine. What you may not know is that the likelihood of becoming dependent on nicotine is also based on the genetics inherited from your parents. Luckily, genetic testing can show whether or not you are genetically inclined to develop a nicotine dependence. A single nucleotide polymorphisms test, or SNP test for short, shows the differences in one nucleotide at a specific location on a chromosome, which can signify the nucleotide variations linked to what you're testing for. Our SNP tests for the 15 genetic variations associated with nicotine dependence. There are 2 genes that are directly linked to nicotine dependence. According to Medline Plus Genetics, one main SNP that has an impact on nicotine dependence is the RS10485057. This SNP affects the OPRM1 gene. The OPRM1 gene makes the mu-opioid receptor. These receptors bind to nerve cells membranes and binds to opioids in the body, triggering pain relief and pleasure. A version of this gene that is associated with a higher likelihood of smoking initiation and nicotine dependence is homozygous G allele. It is still unclear as to how exactly the genotype for this SNP, but in these studies, there was strong correlation between nicotine dependence and the homozygous G allele. Other genotypes like AG and homozygous A are considered normal and do not increase the likelihood for nicotine dependence. It would be useful to include the other 2 variants of this SNP in our test in order to determine if the subject is more likely to become dependent on nicotine. Another SNP we would test for is the RS2302763 SNP. This is associated CHRNB1 codes for an amino acid that makes up a larger protein called neuronic nicotinic acetylcholine receptor. This protein acts as a channel in nerve cells that allows charged ions to pass through the cell membrane when open. Neurotransmitters like dopamine bond to this receptor and open this channel. Nicotine can also cause the channel to open. A study conducted by James McAuliffe and others suggests that the normal CHRNB1 gene is homozygous T. Variants include homozygous C and CT. Variant genotypes homozygous C and CT are associated with an increase in nicotine dependence. It is still unclear as to how exactly the genotype for this SNP functions to cause dependence on nicotine. It would be useful to include the other 2 variants of the SNP to show if someone had healthy, normal nicotine dependence. Both of these SNPs are in genes associated with neurotransmitters or neural activity. These are the same associations responsible for the pleasure and addictive response when nicotine is present, so it's logical that these genes could affect dependence. One protein that is affected is the CYP2A6 enzyme that is produced in the liver. Several SNPs, insertions, deletions, and duplications have been found in this specific gene. We are unsure of how the protein is created, but people with homozygous alleles for the deletion variation had 400% increase high nicotine present in their body compared to those with the normal genotype. This means that if the enzyme is produced incorrectly, it rapidly decreases the body's ability to metabolize nicotine. Without the ability to metabolize nicotine, it remains in the bloodstream and prolongs the effects and exposure of nicotine to the brain. More exposure increases an individual's likelihood to develop a dependence on nicotine. Research has also been conducted on hypothetical risk ratings for people who had SNPs and genes associated with nicotine addiction. Predictive models of genetic risk were created based on prior research from other studies. People having normal genotypes for all of the associated genes, including OPRM1, CHRNB1, and CYP2A6, were given a risk rating of 1.0. People with any one gene affected were given a risk rating of 3.0. People with 2 affected genes rated 9.0. People with 3 affected genes were rated 27.0. Hypothetically, every affected gene increases a person's risk by a factor of 3. Again, because nicotine addiction is a complex disease and influenced by many factors other than genetics, it's hard to quantify exact percentages of risk. Regardless of your genotype, nicotine is a very addictive chemical and affects everyone no matter how they try not to get addicted. Risk ratings are more useful as a general comparison rather than a definite scale. Just because we can perform genetic testing for nicotine dependence does not mean that it will be 100% accurate. There are currently many holes in our information that still is yet to be discovered. It is still unclear how the genotype for the OPRM1 gene, homozygous G, actually functions and therefore increases risk for nicotine dependence. Similarly, we're unsure how exactly the CYP2A6 enzyme is formed and what the function of it is. We just know that it is homozygous and it slows down the metabolization of nicotine. That being said, all the studies referenced showed high correlation with nicotine dependence and those specific alleles. Additionally, even if you test negative for one or all of the traits, you can still develop nicotine dependence. There are many environmental factors that also have an effect on if you will develop a nicotine addiction. If someone is rarely exposed to nicotine for their entire life, they'll never be tempted to start, which doesn't allow for dependence to begin with. Additionally, if someone who tests negative is constantly exposed and wants to smoke or vape to fit in, they can still definitely become addicted. The main ethical concern for this test is the test takers knowing their results. If the results state there is an unlikely chance of genetic predisposition to nicotine dependence, the test taker could be more careless in their nicotine use. When many people quit smoking, they are very proud of themselves because they felt that it was a very hard task to overcome. This makes relapsing and going back to using nicotine more appealing because there is less threat of having to quit and going through the hardships that will come with it all over again. On the flip side, if someone is already addicted to nicotine and finds out that they tested positive, it can make them discouraged from quitting. Knowing that something is going to be very hard may make people shy away from quitting. This could lead to more out-of-pocket costs for the user as they are discouraged to quit. Nicotine dependence is a complex trait that is influenced by both environmental and genetic factors. Regardless of the results of a genetic test, nicotine is highly addictive and none of the consequences come into play if you don't start using it.